Good evening, everyone, and welcome to tonight's webinar. This is our second in our series, all about genetics and regeneration. I'm joined by Dr. Gustavo Torres de Souza, who's leading tonight's education, all focused on epigenetic regulation and gene expression in aging. So the aim is to explore how epigenetic changes regulate gene expression over time and influence biological aging and tissue function. Most of you were here last week as well. If you weren't, then you can catch up. The webinar's on YouTube, so just let us know and we can send you the link. But we'll just jump straight into this second session this evening. And I hope you enjoy. If you do have any questions, please pop them in the Q&A box below, and we'll get through as many as we can at the end, aiming to wrap up by 8 p.m. Okay, over to you, Gustavo.
Everyone. Thank you, the ones who made it to be back to this second session of the webinar. So last week we started in one specific side of genetics, which was kind of the beginning or the beginning of anything we need to know when we're talking about employing genetics in the clinics, but also understanding genetics to understand longevity therapy, longevity in general. Which was the DNA. And now we're moving on in the direction of gene expression. So we started to understand how the genome was encoded, what is kind of physically in the DNA. And now we're going to the second step of our understanding of all that, which is the gene expression per se. So essentially how anything that is written in the DNA becomes a part or becomes a function in the body of our patients. So for us to quickly move on, so we are moving on from the webinar 1 to the webinar 2, in which we're going to gene expression per se. And having that in mind, we have three main topics to cover, which is firstly an introduction on the molecule itself, or the molecular aspect, and some applications of how RNA expression might be present already in your practice. And then we move on to gene expression itself, or the basic part of gene expression, aging, regulation, to finalize, as we can understand that aging as a process of alteration in gene expression too. So these are the main references we have. So the first one is the book. It's the Griffith Introduction to Genetic Analysis, which I mentioned last time. And there are also two new papers I have sent to all of you with a reference material, so that you can kind of have a look at different things a bit more in detail, and newer materials on gene expression. Quick review, we spoke about DNA last time, and one of the topics we spoke when talking about DNA was the usage of nucleotides. So nucleotides, polynucleotides, PDRNs in therapy. And one of the main things is the fact that, well, we have in the backbone of the DNA, so the thing you're seeing here as the backbone of the DNA, we do have the capability of this hydrogen binding between water molecules and the DNA. So this is essentially why we have the capability of using DNA molecules, or the nucleotides or polynucleotides, as a source of hydration. So hydration transdermically, or injectable hydration molecules. So this is one of the usage of DNA. So this means that we're understanding DNA here, when we were understanding DNA here so far physically. And then we also started with a very important topic for rejuvenation and for longevity in general, which is the fact that DNA needs to be duplicated prior to cell proliferation, and that means that DNA gets divided, and we have a machinery that entails the DNA polymerase that goes on over to both sides of the DNA, doing new molecules, or new DNA molecules, and also, which is very important in the longevity aspect, is that cell division and DNA duplication in general requires, and also has in its machinery, the fact that we have proofreading as a a another molecule, so that's the DNA polymerase type 1, that does check if the DNA is being polymerized correctly. So this is another aspect of how DNA is implicated in longevity. And furthermore, replication is controlled. So we have the cell cycle being controlled by external and internal aspects of the cell, and that defines pretty much if this proliferation is happening in a healthy way. And that takes us to finally RNA. So let's say, for instance, that we're applying a certain exosome to a patient. So a patient is being treated with an exosome. Uh, Many of us, I mean, we see loads of brands on exosomes. We see loads of, loads of information on this type of newer therapies. But sometimes it's hard to understand if essentially this has been communicated correctly to us, been communicated correctly to the patients. And one of the aspects on exosomes, or the real exosomes, it's pretty much a known biological concept in which you have molecules or vesicles that are able to bind to our cells. So essentially, whenever we have the fact that we have an external stimulus binding to a receptor in our cells, this information gets transferred to inside the cell. And as it is transferred to inside the cell, one or probably the most important way in which this signaling, which is the transfer from outside to inside the cell, that it does something to our cells, is essentially that when it reaches the cellular target, it will, through the signal transduction, it will generate alterations in gene expression. So essentially what we will learn this in this class will be understanding how and when and what is involved when something comes from out of our cells to inside of it, and it changes how our cells behaves. And so the gene expression is probably the most important aspect of how this happens. So this one is the exosomes. Everyone has heard of it. And you'll see that inside exosomes, so exosomes as therapeutic things, you will see that there are plenty, or we expect that inside of it, we have plenty of different types of molecules. So exosomes, as we know them and as we use them therapeutically, they are aimed, or they are done so that we have this low physical. So we have this little thing with a membrane outside, and it has other types of molecules inside. And inside of them, we do have DNA, might have RNA, but you have also things which are called transcription factors. And the transcription factors are known to be probably the most important type of molecule we have inside exosomes, both our internal exosomes, so exosomes that we produce naturally in our body during some processes, but also exosomes that we use therapeutically in the patients. So what happens is that everything we might have inside a vesicle, they might have a known function or not, or somewhat of a prediction of that function. But the most important thing is that whenever we have vesicles being transferred from one cell to the other, one of the main things that happen is that the content of this vesicle, it gets kind of integrated into a different cell. So this is when it goes to the recipient cell. And some of the main functions that might happen is the fact that we will have alterations in gene expression, and gene expression will mean that we will have different proteins being produced, or proteins being produced at different levels. And this is pretty much how cells change their behavior, and eventually how tissues will differ or become different when we do a certain treatment to this tissue. Well, and this has obviously many, many applications, but I think the things we hear the most are as the usage of exosomes, which will eventually, in the case of what we are discussing here today, is the fact that we have, for instance, exosomes being kind of the carriers of information and carriers of bigger molecules that are able to do things to other cells. So we see many applications of it. So we see many applications on on the skin rejuvenation, the skin longevity, and skin alterations of signs of aging. And what can be important is that, for instance, we can have exosomes carrying collagen signaling, so signals that will make cells express collagen better, for instance. Or we have, for instance, in hair restoration, which is quite a bit of a thing, we have alterations in the beta-catenin pathway that leads to both cell proliferation and function of the hair follicle. We have fibroblast regulation, so cell proliferation due to alterations in gene expression, melanocyte activity, and so on and so forth. So what do I want to tell you about? Why do I want to tell you about this? This is essentially because whenever something comes from outside the cell, so it is a step or a way of doing molecular communication, there's going to be some alteration in either or both epigenetic factors and gene expression, and this will alter the cellular state, and this will lead to tissue and clinical response. So everything we see happening in the patients when we employ certain therapies in them, most of what happens rely on alteration in gene expression. And that takes us to gene expression per se. So the really real topic of today's discussion. When we go to gene expression and epigenetics, I think epigenetics that we hear a lot, and it generates a bit of a both confusion and interest. It brings me one question, and now I will actually stop and wait for someone to answer in the chat, please. The question. So if all cells we have in our body, they have the same genome, so DNA is the same, why are not all cells the same? So we have different cells in our brain, in our mouth, in our heart, everywhere, but they do all have the same genome, they all have the same DNA. Can anyone answer this question, or can anyone speculate why this happens? If not, I'll proceed, but please, if someone can think about it a bit and tell me. There's a chat and there's a question and answer. You can obviously feel free to write if you have an answer. Come on. Not sure if we can talk, but perhaps if you can, it's also fine. Everyone? Okay. So what happens, or what leads us to having the same DNA exactly in all of our cells, or most of our cells, but different cells, is the fact exactly that we do have alterations in cell activity and then gene expression. So what we see here is essentially that. So let's say we have a certain locus in the DNA. It's just the A1 locus. Here, and it is here. Sorry, just erase this one. So the A1 locus, which is a part of the DNA. So let's say we have a whole DNA and we are looking at one part of it. It is present in the cell A. It is present also in the cell A alpha, for instance. What happens? This part here, it does produce the protein A1. So both cells are capable of doing it. However, what happens is that some of these cells might not have the same transcription factors and therefore might not have the capability of producing the same proteins. So what happens here is that if we have some genes which are produced only when this is present, when these transcription factors are present, we have some cells, let's say the cell A has the production of a certain protein on, but both the other cells are not. And this can change, obviously, depending on the cell type and also what stimulus the cell is receiving from the outside or internally. What do I mean by that? Cells, they have the same identity. However, we have a regulation, we have a mechanism of regulation that leads the cells to producing different proteins at the same time. So different genes are going to be expressed, which is the terminology we use. So genes are expressed differentially in different cells in different times. So these are the two things that are most important when we are discussing the use of gene expression or the use of any mechanisms that alter gene expression. Is not all cells produce the same gene expression. So gene expression changes from cell to cell and also changes from time to time. So this is a very important thing. We might be modulating gene expression across time and also modulating gene expression across types of cells. So this, what we are referring to, is inside the central dogma of molecular biology, which we talked about last week, which is essentially the transfer of information from the DNA to the RNA in the transcription process. So what's happening here is essentially that we are getting the information that is in the DNA, and we're doing the first step, the first big step we have into transforming this information into cell activity or cell function, which is essentially then transferring it from DNA to RNA. And when we need to do RNA out of DNA, what happens essentially is that we have a certain places in our genome, in our DNA, in which we have this configuration. I'll show you the simplified configuration below, and then we will see the complex configuration above. So whenever you hear about, well, collagen is being produced or myoglobin is being produced, a certain receptor is being produced by our body, so a certain protein is produced by our cells. What is happening essentially is that we have a certain place in the DNA. So we have the whole DNA in our cells, but we have a certain place in which we have a sequence that we call promoter, which is this sequence here. And we have the coding sequence. So we have the code for that protein right in front of the promoter. And this at least how it works for most of the cells. And what happens? What happens is that in the promoter region, which is what you're seeing below here in this color, so purple color, what happens is that we have a almost set list, almost set sequence of nucleotides that are necessary for what we call the RNase polymerase to bind to the DNA. So it comes to the DNA right in front of where we have a coding sequence for a certain protein, and it's able to anchor itself there and it start producing the RNA itself. So this is pretty much how it happens. So again, the same simplified thing, and we have the promoter, and we have the capability of the DNA polymerase to bind. However, since our cells need to control, and this goes back to the fact that I asked you if all of our cells have the same DNA, why we're not producing all the proteins all the time. What happens is that what differentiates between the cells is the fact that we have other proteins that are called transcription factors that bind to different parts of the DNA, and they more or less organize the DNA so that the RNase polymerase or the RNA polymerase can come to the DNA and start doing its job of actually producing RNA. So this is the most important slide and the most important information you need to have in your minds whenever you are thinking about producing or an alteration in a patient, or thinking about devising a certain therapy that is going to lead to alterations in the functioning of a certain tissue, just as we said about the exosomes, but that also applies to obviously any therapy you are devising in the patient. So as RNA is produced, so this is essentially how it works or how it stays in our cells. So as we have spoken or discussed last class, we'd have our DNA being the storage way or the storage molecule for information. And then we have our transcription happening, which is essentially what we just spoke about. So initiation of transcription depends on the binding of the RNA polymerase to the DNA, and it goes on to producing the RNA, processing this RNA. So this is also the RNA is capable of, in the end, producing the protein, and in the end, the abundance or the amount of RNA that we have in certain cells, which is essentially when we have the RNA coming from the nucleus to the cytoplasm. This is where essentially RNA has its function. So first thing to have in mind is for RNA to be produced, or for a certain gene to start being part of the cell function, we need the transcription factors to organize the DNA tridimensionally so that the RNA polymerase 2 can bind to it. And then when it binds to it, it produces RNA. When it produces RNA, essentially RNA goes to the cytoplasm and it has its function, which is the production, in most cases, of a protein. And here is our last information, or a last aspect on how essentially um, this is capable of altering the behavior between cells, which is what we call alternative splicing. So this is the conversion or the processing of RNA readily produced from the DNA so that it's fully functional. So the DNA or the gene is transcribed into a precursor RNA. So let's say we have a whole chunk of the DNA in a certain position of our genome, and it has information to produce proteins that are suitable for many types of cells. What happens here is the following. We produce all of that, so it's the primary transcript or the precursor RNA, and some different cells use different parts of this. Let's say the striated muscle uses the part 1, 2B, 3, 4, and 5, while the smooth muscle might use the 1A, the 2, the 3, 4, 5, and so on and so forth. So this is the second aspect on how different cells behave differently because of RNA expression. So RNA expression entails the production of RNA, and also this little differences that we have in the final RNA molecule that will lead, as you can see on your right, to alterations in essentially how proteins differ in different cells. So for instance, we have here this receptor, the type 1 and the type 2, just as we have, for instance, the 5-alpha reductase enzyme that we use normally for finasteride and dutasteride. This is essentially a receptor that differs only in a small bit. So let's say one of this receptor uses one of the parts of the RNA and the other one uses another part of the RNA produced. So this is the second way cells differ when or cells become different due to gene expression. Well, and for us to kind of finalize the notion of gene expression, I would like to show you one kind of real life example of how this happens. So we know very much that inflammation or the inflammatory process is very important for both our cells to respond to foreign agents, but also to be also it needs to be controlled in view of loads of processes used therapeutically in the aesthetics, but also in regeneration and restoring of function of the body in general. What do I mean by that? Let's recover the fact that we have a certain part of the DNA where we have the promoter and the coding region, in which we have the possibility of the transcription factors organizing the setting, and then the RNA polymerase binds to a certain place, and we produce the RNA or produce the proteins. That's very good. However, how can this be important? How can this be relevant? So If something comes from out of our cells and binds to a receptor in our cell, and it leads to alteration in the quantity of transcription factors or the function of transcription factors, this is what is going to lead to alterations to timely alterations or alterations that happen across time in our cells. What do I mean by that? So I told you that one of the most important things about gene expression is that it changes across time. So let's say we have a cell of our immune system that is normally not producing inflammatory profile protein. So it's not normally producing interleukin type 1, or necrosis factor, and so on and so forth. So normally in a homeostasis state, our body wouldn't be producing anything that is related to inflammation. So it produces it in a balance, but normally we're not inflaming our body. So this is a normal behavior of a cell. At least this is um, initially like that. However, let's say our cell has entered in contact with a certain external factor. The very known one is the LPS. So it binds to a certain receptor, like the toll-like receptor 4. And when this receptor um, transduces or takes the information from outside the cell to inside the cell, we all learned in the past that, well, this leads to inflammation. But how does it lead to inflammation? Is the fact that this leads to alteration in the NF-kappa beta transcription factor and the IRF transcription factor. And what they do is that essentially they bind to the DNA, just as we know here, and it leads then to alterations in gene expression. So as it leads to alterations in gene expressions, we have many alterations happening in those cells, production of the molecules related to inflammation, and therefore the whole tissue becomes inflamed. So this is one real life example here. I would like you to internalize that everything that is happening in our body, or most of what is happening to our tissues, happens because the cells are altering how they are expressing certain genes. So we have loads of processes happening. And obviously, you all know that if you want to block inflammation, for instance, you would use a corticoid. A corticoid, what it does is that it goes directly inside the cell. And one of the main activities really is blocking the uh, function of the NF-kappa beta into really producing the primary response of inflammation. So this is a pretty important thing for you to understand the dynamics between treatment and also tissue response. So tissue response, most of the situation happens through gene expression. And then we have the other side of gene expression, and probably the best known, or what we hear the most about gene expression, which is epigenetics. Epigenetics is something we hear a lot. So we hear a lot about epigenetics. It's something that, it became part um, of discussions in the longevity sector, in the aesthetics world, and so on and so forth. But no one really knows what, or most of people discussing this might not know what epigenetics is. And epigenetics is nothing different from another part of the gene expression control. So we have transcription factors controlling gene expression. As we see, it organizes the DNA. And we have epigenetics or epigenetic alterations also helping us control how genes are expressed, or also controlling how RNA is produced from the DNA. So if we take a look at how, sorry, at how DNA is organized, so we tend to think as DNA as a linear thing. However, in our cells, specifically in the eukaryotic, or in the cells we have in the human body, DNA is not a straight line of just nucleotides. So when we talk about nucleotides in our body, they are not just a sequence. This sequence is really wound around certain proteins that are called histones. And this forms what we call nucleosomes. Doesn't matter. But what essentially happens is that the DNA is all turned around itself and around the histones. And why is this important? This is important because, as you can see, we could think of this chunk of DNA here being always available, being always in a line. But what actually happens is that in most of the situations, this same, let's just reproduce it here, change colors. If this part of the DNA is here, what normally we would think is that, well, this is always in a line. I can always read that part of the DNA. But what actually happens is that in most situations, what happens and what controls the functioning of our cells is that we have this chunk of DNA organized around a protein called histone, which is actually our protein that is composed of different ones, but it doesn't really matter. So this is what composes what we call the chromatin. So the chromatin is this physical organization of our DNA. So our DNA is really always like that. It's always turned and compacted into a certain structure around the histones. Why is this important? This is important because all these factors we have seen below or previously, which is the enhancer, the promoter, the gene itself, and so on and so forth, they might be not accessible because we have them around the histone. So what happens is that when we need a gene to be expressed, we need them to be made available. So this is what happens in our epigenetics alterations. So epigenetics stands for alterations happening on the DNA. So epi is on the DNA. So these alterations are happening on the histones and also on some of the nucleotides, but it's a different story. So what happens is that the epigenetic alterations or the epigenetic markers, they are done or written on the DNA, and they can be erased, of course, so that then when the reader, which is the RNA polymerase we have discussed about, it will depend whether this reader can or cannot read the DNA that has this marker on. And obviously, we have many types of other alterations, and they might be either around promoters or inside the gene or between genes. But what is more important is that in order for us to have gene expression, we need the histones to be slightly less compacted so that then the RNA polymerase can come inside it and inside this low part of the DNA and do its job of producing RNA. So RNA as gene expression, as we have spoken before. So you will see that, and as a summary of everything from this basic part, is the fact that anything we do and any alteration in the body, any alteration, either therapeutic alteration that happens from one side, so we are doing a therapy and we want the tissue to do something, or when the tissue is doing something and we want to recover this tissue to a certain state, it always comes down to a certain signal happening from outside the cell in most situations. It is transduced inside the cell. We have alteration in transcription factors. So the transcription factors are able to organize the DNA. They also change chromatin accessibility, and this is what we call epigenetics. So whenever again you hear about epigenetics, this is what we are talking about. It changes RNA expression, so how much RNA is produced, and therefore how the cells behave. Well, so all of that is very theoretic. All of that is very much related, to how our cells function, to how our cells um, change across time. However, we also have the understanding that both therapeutically in longevity, but also following patients across time, um, gene expression changes also are involved in um, how aging happens. So I told you that probably the most important things about gene expression is that it changes across cells and it changes across time. So it is very pretty much expected that uh, as we age, so as time passes, we would expect gene expression to change in certain cells in a certain way. So there are some markers of gene expression that will be changed across time depending on gene expression. So some behaviors of our tissues, or some behaviors of how, or some ways our tissues behave, will change across time, but across our age or aging process as well, due to alterations in gene expression. And that is really what I want for us to have as a final information here. So let's say we have again that schema in which we had the promoter, so a certain region to which the RNA polymerase binds, and this produces, a certain protein. So when the RNA polymerase passes here, we have RNA, and we have a certain protein being produced. Very good. Uh, what um, what happens from that is that there are some markers that when someone is young, they will be very strongly producing the RNA that is in front of here, but very weakly producing what is in front of this place. So this is the dichotomy. So when someone is young, the idea behind it is that there are some places of the DNA in which young people produce very much of a certain protein, but then less of a certain other protein, and old people will produce elderly people will produce less of a certain protein, but then as they grow older, they will produce more of a certain protein. So this is the idea of alteration in expression pattern across time related to longevity. We have seen this expression variability happening in many situations, and also tissue specific. And this is pretty much a very complicated graph to be interpreted. But what I want you to take from this, and also obviously if you want to kind of screenshot to see this paper later, what happens here is that we have certain tissue, or some tissues are differing in how much we produce certain proteins across time. So we do have tissues in which we have very much difference, as we have, for instance, in the case of blood, which also is very important, because blood cells do suffer some mutations across time. So also a different story. But what is more important here is that all across our tissue, we have both variations in expression across time, and also variation of expression across tissue, but also we have variations in expression across time of our lives, so years lived. So we do have a difference in gene expression as we grow old. So all we saw before, which is the binding of the RNA polymerase, the production of RNA, production of a protein, this changes dramatically in some cases as when we grow from younger individuals to older individuals. So obviously we do have some types of cells that differ less as the liver and muscle, surprisingly, but some cells do have quite a bit of alteration, such as the blood cells, as we grow old. Well, a different thing, but also related to expression, is the fact that there's alterations in expression pattern in inflammatory profiles and also DNA repair genes. So this is the our second part of how gene expression changes across time. It's also the fact that, as we have discussed previously, and just seen that, for instance, blood cells do differ too much across time in terms of expression. Some very important proteins differ very much when aging. And this is pretty much marked when we talk about inflammation. And this is probably why in the later year, the terminology inflammaging has appeared very often in the longevity discussion. It's really because inflammation is something that leads to cell proliferation, to alterations in tissue function, and so on and so forth. And we know that as we grow old, there is pretty much an alteration in how we control inflammation. and also how we respond to both, alterations in cell and also alterations in response mechanisms are related to inflammation. And why is this important? When we have alterations in the expression of inflammatory proteins and also repair genes, this is why we will have less control of cell proliferation and also less control of inflammation and control in tissue repair when we grow old. This also means that this can very importantly be a focus for therapeutically use of longevity therapy. So the use of stem cell exosomes, as have seen in the beginning, of specific markers, specific things, they will all lead us. The aim of all that is to keep the expression or to normalize the expression across aging as a longevity factor. And we have something very interesting to see in the end. Um, as expression might change simply due to alterations in the overall cell signaling, we also have the fact that epigenetic changes happen across time. So, very important genes related to aging and also related to cell proliferation, they become more methylated, so they receive epigenetic markers that lead them not to be produced very much, and this is very important. So as epigenetic passes from young to older, we have some proteins related to cell proliferation and cell control and also repair of DNA being left expressed across time. And the same thing goes again to inflammatory profiles. So the aging tissue also change very much the epigenetic factors that lead to control of uh, um, of inflammatory profiles. Finally, we come to the last portion of this, which is what I wanted to show you on the the kind of final interest of all that. And I'm going to use the skin to exemplify this, but I want you to bear in mind that the idea is that the interaction between the outer world and our cells, it pretty much always passes through gene expression. So I'm showing you here a bit on the cutaneous exposome. So exposome is what we hear a lot as well, which is everything that is outside our cells and might interfere with the functioning of our cells. And we have pretty much many factors happening, or many processes happening across time in our cells. And this is now specifically for aging of the skin, but I want you to bear in mind that this is true for all the tissues. So as the skin and as the cells are exposed to certain factors, what happens is essentially that, well, we have DNA damage. So this is from last class. We'll see this again when we talk about aging. However, we have many other aspects of it, which are essentially alterations in gene expression. So we have senescent cells with alterations in gene expressions. We have alterations in the inflammatory or immune function and endothelial function happening due to the aging and alteration in gene expression in these aspects. And why is this important? This is important because this is also what we can use in order to devise therapies and to employ these therapies into patients. Obviously, it would be ideal if we could use therapies that very much go on to specific factors and lead to alterations in gene expression, such as still in studies, analytics that go directly to certain expressions, certain things, or cell reprogramming, epigenetic reprogramming. All of that is very interesting, but however, so far this is not something we can do very specifically, like stem cell therapy. All of those things, they are partially not possible to be done. Perhaps with the exception of the usage of exosomes or extracellular vesicles from stem cells, because they have been employed already therapeutically. However, My aim here is to show you that, well, gene expression can be changed in many situations by known drugs and known aspects of the longevity arsenal we have so far. And some of them are widely known and also widely important, such as metformin and rapamycin. These are repurposed drugs that change, in the case of metformin, the production of the AMP kinase, and the rapamycin changes the expression of the mTOR function, as well as we have also the NAD precursors, also changing how DNA is repaired, but most importantly, how all the expression of the DNA repair enzymes and the DNA production enzymes happening in our bodies. So what I wanted to leave you here as a message is that understanding gene expression happens to also help you understanding how we can therapeutically approach patients and why we approach patients therapeutically in order to improve their overall behavior or the tissue behavior and to promote longevity. So finally, so that you can have this as a final thing, um, regenerative signaling, so things that happen outside ourselves, they rely on pre-existing mechanisms inside our cells. And these mechanisms, they're essentially gene expression alterations, which might happen directly through signaling. So a receptor changes gene expression. But in most situations, or in many of the situations, this also happens through the histone modifications and chromatin remodeling, which is what we call epigenetics. So whatever we are doing to our patients to improve their aging and to promote longevity, it will depend and rely heavily on what we are telling ourselves to express differently. And as we have seen already, and this leads us to the questions, is the fact that many of these things altering are cell proliferation and inflammation. So this also goes back to all the attempts we have in longevity. They will at some point lead to alterations in gene expression, but focused on inflammation, so keeping inflammation under control, and also keeping cell proliferation, as we've seen in the previous class as well, in control. I would like to open us now for questions and also well communicate to all of you and see if you have anything to say.
Thank you so much, Gustavo. That was another excellent presentation in our genetics series. Does have anyone have any questions? We haven't had any popping up just yet, but pop them in the Q&A at the bottom of your screen if you do. Give you a couple of minutes. But yeah, thank you very much. That was so interesting. If there was sort of one key takeaway point you would advise people leave with this evening, what would it be?
Yeah, I'd say that the main takeaway point here is that we do have mechanisms already in our cells that we can explore in the overall goal and aim of therapeutically changing patients in the longevity market. And probably gene expression is the main central one because it is involved in just everything in our bodies. So understanding gene expression always will make you a better prescriber, a better um, better therapeutically, better clinically, in order to find ways to improve your patient's overall function.
Yeah, absolutely. Thank you so much. So we haven't had any questions this evening. If anything pops up between now and next week, then do just send us a message, info@raminstitute.com. We'll be here next Wednesday, same time, 7 o'clock. You can watch this webinar back and you can watch last week's back. If you missed
I
that,
just
please let us know and we can
just
send you the link.
saw just saw a question here from Dr. Joya. We have always been accepting that good inflammation was the basis of aesthetic medicine improvement. Does it exist, good inflammation? Would it be better to be able to switch off gene expression? This is a very important question and also very challenging. The best answer to this question is there is not good inflammation, but there is good management of inflammation. So essentially, what happens is that, well, controlled inflammation is the basis of aesthetic medicine, as you said. This is a very important remark. However, this inflammation always must be controlled, and this control always passes through gene expression. Sometimes, what happens, for instance, let's say you do laser or you do microneedling, there's always a step further that if you cross that step, the gene expression related to inflammation is going to be so high that the gene expression related to inflammation control is not going to be able to, in a timely manner, surpass it and avoid tissue remodeling from happening in a bad way. And you all know that. We all have seen this because in some situations, some patients do use injectables or, uh, um, energy devices or microneedling and so on and so forth. What can happen is that if gene expression of genes related to inflammation are too high, what will happen is that it's not going to be controlled in time, and therefore you will have fibroblast proliferation too fast that will lead to scar tissue formation, to fibroblast or to fibrosis, and so on and so forth. So, does good inflammation exist? I wouldn't say that good inflammation exists, because inflammation happens for a reason. Sometimes it's good simply because it's controlling a certain process. But I would say that controlled inflammation is the good inflammation. Second question, would it be better to be able to switch off expression, gene expression? And I assume, if you could perhaps give me a follow-up to that question. I assume you're talking about switching off gene expression in of inflammatory genes, right? I thank you. But just to finalize your question, when you mean to being able to switch off gene expression, you mean on genes related to inflammation, right? Okay, very good question. So this is essentially what we would do if, for instance, you apply a corticoid while doing a certain therapy. What you are doing effectively is trying to switch off, or at least to control gene expression of the inflammatory profile proteins. So this is exactly what is being done and what exactly needs to be done in some cases to control inflammation so that controlled inflammation can be kept as good and therapeutically correct. Does it make sense? Thank you very much. Obviously, this doesn't mean that we always use corticoids or always control inflammation. Um, the how is essentially because when corticoids are used, they are going to be the ones to, alongside the NF-kappa beta we've seen during our conversation, alongside with those, they're going to also increase the expression of proteins or interleukins related to inflammation control, such as the IL-10, for instance, that controls inflammation. It is not always the case. However, what really needs to be put into consideration here is that sometimes we need to control inflammation. Sometimes we can need to control inflammation externally. Sometimes we just do the damage to a certain tissue more controlledly, and therefore the inflammation doesn't surpass what our system can control in terms of gene expression. Thank you. Thank you.
Hopefully,
Thank you very
that
much.
answered all the questions. Thank you so much, Gustavo, and everyone for joining. Like I
Thank you.
said, you can watch this back. The link will be on our website soon. You can also register for Ramsey, save £25 in July. So it's 125 plus VAT at the moment. Just head to our website. That'll be November the 7th. Thank you very much. Look forward to seeing
Thank
you
you.
all again next week.
Thank you all very much.
Bye
See you
bye.
all. Bye bye.